Hello students and my dear friends, myself Madhu Vancha, Assistant Professor of Microbiology. In this session, we'll discuss some of the important microbiology MCQs which are helpful for you in the upcoming examination, that is TGSPSC or like other medical recruitment board examinations. Question number 1. What moderant is used in the gram staining? Option A, crystal violet. Option B, safranin. Option C, acid alcohol. Option D, iodine. So here the right answer is iodine. So crystal violet is the primary stain and safranin is the counter stain. Acid alcohol is the decolorizer and iodine is the moderant stain. Question number 2. All of the following are motile bacteria except option A, E. coli, option B, Klebsiella, option C, Vibrio, option D, Proteus. So the right answer is Klebsiella. If you see E. coli, so E. coli, it has the peritrichous flagella, which means flagella present all over the cell. And Vibrio cholera has the darting motility or vibratory motility. And Proteus is also highly motile organism. Only Klebsiella is non-motile. Next, question number 3. The coagulase test is done to differentiate. Option A, Staphylococcus aureus from Staphylococcus epidermidis. Option B, Staphylococcus epidermidis from Neisseria meningitidis. Option C, Streptococcus pyogenes from Enterococcus. Option D, Streptococcus pyogenes from Staphylococcus aureus. So the right answer is Staphylococcus aureus from Staphylococcus epidermidis. So coagulase test, it is done to differentiate Staphylococcus aureus from Staphylococcus epidermidis. Staphylococcus aureus, it is a pyogenic organism. It causes pus-forming infection. Staphylococcus epidermidis, it is a commensal. It is an opportunistic infection. It causes infections in the patients with the artificial heart valves. Next question. Which of the following parasite is found in the urine specimen? Option A, Taenia species. Option B, Trichomonas vaginalis. Option C, Trichura. Option D, roundworm. And the right answer is Trichomonas vaginalis. Here, Trichomonas vaginalis, it is the only parasite which causes STI, sexually transmitted infections. So the right answer is Trichomonas vaginalis. Next question. Which of the following is a gram-negative bacilli? Option A, Staphylococcus aureus. Option B, Staphylococcus citrus. Option B, Streptococcus pneumoniae. Option D, Pseudomonas aeruginosa. So here the right answer is Pseudomonas aeruginosa. So for this, the answer, identification of answer is easy. Here you can see Staphylococcus, and Streptococcus. So here in the name itself, it suggests like it is a cocci. Only Pseudomonas aeruginosa is the bacilli. It is a rod-shaped bacteria. Next question. Comma-shaped bacteria are known as A, Staphylococci, B, Streptococci, C, Vibrio, and D, Bacillus. So here right answer is option C, that is Vibrio. Next question. A positive tuberculin skin test is indicated by an area of induration. Option A, less than 5 mm in the diameter. Option B, 6 to 9 mm in the diameter. Option C, 10 mm in the diameter. Option D, no induration. The right answer is option C, that is 10 mm in the diameter. So tuberculin skin test, it is based upon the delayed type of hypersensitivity. So after 48 hours or after 72 hours of examination, if the induration is more than 10 mm or 10 mm in the diameter, is the suggestive of the tuberculosis. Next question, diagnostic test used for tuberculosis is A. Montu test, B. Dick test, and C. Chick test, and D. Vidal test. So here the right answer is option A. Montauk's test. Next question, acid fast staining is used to detect option A. Mycobacterium tuberculosis, option B. Clostridium tetani, option C. HIV or AIDS, option D. Meningococci. So the right answer is option A. Mycobacterium tuberculosis. So Mycobacterium tuberculosis, it is also called as acid fast bacillus. Acid fast staining, it is also called as Ziehl-Neelsen staining or AFB staining. Next question, method of sterilization used for culture media is option A. Autoclave, option B. hot air oven, option C. Pasteurization, option D. None of the above. So here the right answer is option A. Autoclave. Autoclave, it works based upon the principle of steam under pressure, or that is above 100 degrees centigrade. In the autoclave, the culture media, it is sterilized at 121 degrees centigrade, 15 lb pressure for 15 minutes. Hot air oven is based upon the dry heat, and pasteurization, it is a process of killing of pathogens which are present in the milk. So that we use like temperature less than 100 degrees centigrade. Next question, shape of cocci, A. rod shape, B. spherical, C. comma shape, and option D. spiral. The right answer is spherical. Next question, VDRL test is used in the diagnosis of option A. syphilis, option B. HIV, option C. typhoid, and option D. tuberculosis. The right answer is syphilis. VDRL test, Venereal Disease Research Laboratory. This laboratory is located in the New York. So this test was developed first time in that laboratory. So that's why it is named as VDRL laboratory. So it is helpful in the diagnosis of syphilis, which is caused by the spirochete, that is called like Treponema pallidum. Next question, temperature range used for autoclaving is option A. 100 degrees centigrade for 10 minutes, option B. 100 degrees centigrade for 30 minutes, option C. 121 degrees centigrade for 15 minutes, and option D. 121 degrees centigrade for 30 minutes. Here the right answer is option C. 121 degrees centigrade for 15 minutes. So this is the holding time. As I told you, autoclaving, it works based upon the principle of steam under pressure. It comes under temperature above 100 degrees centigrade, that is under moist heat methods of sterilization. Next question, presence of red blood cells in the urine is referred to as option A. hematuria, option B. hemoglobinuria, option C. anuria, option D. chyluria. The right answer is option A. hematuria. So hematuria means presence of RBC in the blood. Hemoglobinuria means presence of hemoglobin in the urine. And option C, anuria, that is absence of urine. And D is chyluria. Chyluria means milky color urination. Next question, the father of bacteriology is option A. Anthony van Leeuwenhoek, option B. Edward Jenner, option C. Robert Koch, option D. Rudolf Virchow. Here the right answer is option C, that is Robert Koch. Next question, Vidal test is used for diagnosis of option A. anthrax, option B. typhoid, option C. chickenpox, and option D, Vidal test. Sorry, option D, syphilis. The right answer is option B, typhoid. Next question. Person who harbors a pathogen and not suffering from any symptoms. Option A, vector. Option B, symbiont. Option C, carrier. Option D, reservoir. Here the right answer is option C, that is carrier. So what is vector? Vector, it is an arthropod, such as mosquitoes, houseflies, sandfly, all these flies, they comes under vectors. They are the arthropods. And the reservoir, which means which harbors the like a pathogen, that is called as like a reservoir. So symbiosis or symbiont, that is a mutual relationship between the like two organisms. Next question. The instruments used for sterilization by dry heat. Option A, autoclave. Option B, vaccine bath. Option C, Koch-Arnold steamer. Option D, hot air oven. Here the right answer is option D, hot air oven. Hot air oven, it works on the dry heat. It comes under dry heat. What is the holding time of hot air oven? That is 160 degrees centigrade for 2 hours. So here, autoclave, vaccine bath, Koch-Arnold steamer. So these three instruments comes under moist heat methods of sterilization. Next question. The selective media used for tubercle bacilli. Option A, SS agar. Option B, LJ media. Option C, RCM. And option D, CA. And here the right answer is option B, LJ media, that is Lunsten-Jensen media. So Lunsten-Jensen media is the selective media. It is for tubercle bacilli. What is the selective media? Selective media is the solid media which has added inhibitory substances which suppress the growth of unwanted bacteria and they promote the growth of like a desired organism or wanted bacteria. So LJ media, it is a solid media. And remember one point, LJ media is the only solid media which is prepared without adding agar-agar. Here, in the preparation of LJ media, we add egg. Egg acts as a solidifying agent. So here the right answer is LJ media. And if you see SS agar, that is Salmonella, Shigella agar, that is selective medium for isolation of Salmonella and Shigella. And RCM or RCMB, Robertson cooked meat broth, that is for growing of anaerobic bacteria. It supports the growth of anaerobic bacteria. And CA, that is chocolate agar. Chocolate agar is not selective media. Chocolate agar is enriched media. Next question. The test used in blood bank to detect sensitizing antibodies. Option A, Papin test. Option B, albumin test. Option C, saline test. Option D, antiglobulin test. The right answer is option D, antiglobulin test. Next question. The bacteria appears in Chinese letter pattern. Option A, Salmonella. Option B, Corynebacterium diphtheriae. Option C, Streptococcus. Option D, Staphylococcus. So here the right answer is Corynebacterium diphtheriae. If you see Salmonella, Salmonella is the gram-negative bacillus, that is the rod-shaped bacteria. Streptococcus, it is the cocci, it is arranged in the chain form. Staphylococci, the cocci is arranged as clusters or grape-like bunches of grapes. So here the correct answer is Corynebacterium diphtheriae. So Corynebacterium diphtheriae can be identified by Albert staining. So in the Albert staining, this bacteria appears as apple green colored bacilli with bluish black metachromatic granules, which are arranged in the Chinese letter pattern or cuneiform arrangement. Next question. The scientist discovered virus. Option A, Alexander Fleming. Option B, Dmitry Ivanovsky. Option C, Louis Pasteur. Option D, Robert Koch. And the right answer is Ivanovsky. Next question. Example for a protozoan parasite. Option A, Taenia species. Option B, Ascaris lumbricoides. Option C, Plasmodium. Option D, Enterobius vermicularis. The right answer is Plasmodium species. Here, Taenia species such as Taenia saginata and Taenia solium, they are the cystores. Ascaris lumbricoides, it is a nematode. Enterobius vermicularis is also a nematode. The right answer is Plasmodium, that is a protozoa. Next question, the bacteria known as Hansen's bacillus. Option A, Mycobacterium tuberculosis. Option B, Mycobacterium leprae. Option C, Lactobacillus. Option D, Mycobacterium intermedius. Here the right answer is option B, Mycobacterium leprae. Mycobacterium leprae, it was discovered by the scientist Hansen's. That's why it is named as Hansen's bacillus. And the leprosy, Mycobacterium leprae causes leprosy. Leprosy, it is also called as Hansen's disease. Next question, darting motility is shown by option A, Staphylococcus. Option B, Vibrio cholera. Option C, E. coli. Option D, Pneumococcus. Here the right answer is option B, Vibrio cholera. Here, if you see Staphylococcus and Pneumococcus, these two are gram-positive cocci. So here remember one point, all cocci are non-motile. And E. coli is also motile, but it is having the like a peritrichous flagella. Vibrio cholera has single polar flagella. It has characteristic vibratory motility. Okay, then Vibrio cholera, it has characteristic vibratory motility. The word Vibrio is derived from the vibratory motility. So it has darting motility. Next question, example for a transport medium. Option A, TCBS medium. Option B, BHI medium, that is brain heart infusion. Next question, next option, option C, that is VR medium. Option D, SDA medium. Here the right answer is option C, VR medium. VR medium means Venkatraman Ramakrishnan medium. This is a transport medium for transporting the like Vibrio cholera. And TCBS medium, that is selective media for Vibrio cholera. And if you see BHI, brain heart infusion agar, and SDA medium, Sabrats dextrose agar medium, these two culture media, they supports the growth of the fungi. Next question, which of the following is a gram-positive bacillus? Option A, Salmonella. Option B, Clostridium. Option C, Shigella. Option D, Proteus. Here the right answer is option B, Clostridium. Salmonella, Shigella, and Proteus. These three bacteria are gram-negative bacillus. Clostridium tetani, Clostridium botulinum, and other Clostridium species, they are gram-positive bacillus. Next question, Bombay blood group was discovered by option A, Deci. Option B, Bende. Option C, Winthrop. Option D, Landsteiner. The right answer is option B, Dr. Bende. Next question, Trichuris is commonly called as whipworm because option A, it is found in whip. Option B, eggs are whip-shaped. Option C, larva are whip-shaped. Option D, adult worm is whip-shaped. The right answer is option D, adult worm is whip-shaped. Next question, the largest immunoglobulin. Option A, IgG. Option B, IgM. Option C, IgE. Option D, IgD. The right answer is option B, that is IgM. IgM is the largest immunoglobulin. IgM, it is a pentamer, which is made up of 5 monomeric units. That is the largest immunoglobulin. Next question. The antigen popularly called as Australian antigen. Option A, tetanolysin. Option B, malarial antigen. Option C, pneumococcal antigen. Option D, hepatitis B, surface antigen. Here the right answer is option D, hepatitis B, surface antigen. Next question. Pregnancy test is based on detection of option A, human chorionic gonadotropin hormone. Option B, estrogen. Option C, prolactin. Option D, progesterone. Here the right answer is option A, human chorionic gonadotropin. Next question. Example of an anti-tuberculous drug. Option A, salicylates. Option B, penicillin. Option D, acyclovir. Option D, isoniazid. Here the right answer is option D, isoniazid. Next question. The infections transmitted from infected animals to man. Option A, zoonotic disease. Option B, exotic disease. Option C, contagious disease. Option D, nosocomial disease. The right answer is zoonotic disease. So what is meant by zoonotic disease? The infections which are transmitted from animals to humans. They are called as zoonotic infections. Example, rabies, toxoplasmosis, and many of the parasitic infections and plague disease from the rats and Mycobacterium bovis, that is from the infected cattle to humans. All these are zoonotic infections. Next question. His staining is used for demonstration of option A, bacterial spore. Option B, bacterial capsule. Option C, fungal mycelia. Option D, fungal spore. So the right answer is option B, bacterial capsule. Next question. Gamma interferon is produced by option A, Kupffer cells. Option B, monocyte. Option C, lymphocytes. Option D, macrophages. Here the right answer is option C, lymphocytes. So what is meant by interferon? Interferons, they are the glycoprotein molecules. They have the antiviral activity. They work against the viral infections. Next question. Example for RNA viruses. Option A, herpes virus. Option B, adenovirus. Option B, picornavirus. Option D, papavirus. The right answer is option C, that is picornavirus. Herpes virus, adenovirus, and papavirus, they are DNA viruses. They consist of DNA as genetic material. Next question. The stain used for negative staining. Option A, Indian ink. Option B, neutral red. Option C, carbamazepine. Option D, methyl violet. The right answer is option A, India ink. So India ink, it is used in the negative staining. So negative staining, it is especially helpful for identification of capsule. Next question. Example for an autoimmune disease. A, diphtheria. B, rheumatoid arthritis. Option C, tetanus. Option D, kala azar. The right answer is option B, rheumatoid arthritis. Rheumatoid arthritis is autoimmune disease. Diphtheria, tetanus, and kala azar, they are not autoimmune diseases. They are not autoimmune diseases. Next question. The full form of NIH swab is option A, National Institute of Hematology. Option B, National Institute of Histology. Option C, National Institute of Hormones. Option D, National Institute of Health. The right answer is option D, National Institute of Health. That is NIH swab. National Institute of Health swab. Next question. Otitis media is an infection related to option A, eyes. Option B, nose. Option C, mouth. Option D, ears. The right answer is option D, ears. Next question. The destruction of all microorganisms, including spores, is called as option A, sanitation. Option B, sterilization. Option C, disinfection. Option D, antisepsis. The right answer is sterilization. So sterilization, it is a process of killing of all microorganisms, including the spores. And disinfection means it is the destruction of all pathogenic microorganisms, but not necessarily the spores. Next question. Which factor would interfere with the growth of a pathogen? Option A, alkaline pH. Option B, acidic pH. Option C, darkness. And option D, moist environment. The right answer is option B, acidic pH. Acidic pH, generally it interferes with the growth of the pathogen. It affects the growth of the bacteria. Next question. When a agar plate is streaked correctly, the final result is option A, individual colonies. Option B, no growth of pathogens. Option C, dense growth which covers the plate. And option D, a pure culture. The right answer is option A, individual colonies. Next question. What solution is used to clean up biohazardous spills on the floor? Option A, Dettol. Option B, Lysol. Option B, 1% hypochlorite. And option D is a spirit. The right answer is option C, that is freshly prepared 1% sodium hypochlorite solution. So sodium hypochlorite solution, so it is used in the like spill management. If any accidental spills happens, in that cases, the spillage area should be covered by 1% hypochlorite solution. That is for minimum of 20 minutes. Next question. Which of the following is associated with type 1 hypersensitivity? Option A, IgA. Option B, IgE. Option C, IgM. Option D, IgT. The right answer is option B, that is IgE. So IgE immunoglobulin that mediates type 1 hypersensitivity, that is anaphylaxis and atopy. IgE antibody, it is also called as reagent antibody. Next question. Which is true regarding Ziehl-Neelsen staining except option A, it is also called as acid fast staining. Option B, carbamazepine, acid alcohol and methylene blue are used. Option C, it is done to detect tuberculosis. And option D, the mycobacterium stains blue. And here the answer is option D, that is mycobacterium stains blue. So this mycobacterium stains blue is wrong. So Zn staining, it is also called as acid fast staining. We use carbamazepine, acid alcohol and methylene blue. These three reagents are used in staining, and it is helpful to identify the mycobacterium tuberculosis. Then what is the result? How the bacteria will appear here? The mycobacterium tuberculosis appears bright red in the color against the blue background. So this blue background, they are pus-and epithelial cells. So this bacteria appears bright red in the color, the background is blue. So here the answer is option D, mycobacterium stains blue. Next question. Who introduced concepts of antiseptics? Option A, Robert Koch. Option B, Louis Pasteur. Option C, Joseph Lister. And option D, Antony van Leeuwenhoek. The right answer is option C, Joseph Lister. Next question. Viruses are best grown in option A, enriched media. Option B, nutrient agar. Option C, liquid media with a serum. And option D, culture cells. Here the right answer is option D, culture cells. Remember one point, viruses, they can be grown only in the living cells, that is in the tissue cultures or embryonated eggs Okay, like that in the tissue cultures only the viruses can be grown. If you see enriched media, nutrient agar, and liquid media with the serum, all these are artificial culture media. They supports the growth of bacteria only, not the viruses. Next question. The most common cause of UTI is urinary tract infections is option A, E. coli, option B, Bacillus subtilis, option C, Streptococcus pyogenes, option D, Klebsiella pneumonia. The right answer is option A, E. coli. E. coli is the most common cause of urinary tract infections. Next question. First antibody to appear after stimulation by an antigen is option A, IgM, option B, IgA, option C, IgE, and option D, IgG. The right answer is option A, IgM. So IgM appears early in response to the infection, but the half-life of IgM is only 5 days. So after that, IgM, it is replaced by IgG antibody. So presence of IgM antibody indicates recent infection, and presence of IgG antibody indicates the past infection. Next question. Universal precautions came into existence in the year. Option A, 1990. Option B, 1985. Option C, 1987. And option D, 1980. The right answer is option B, 1985. Next question. Glutaraldehyde is an example of option A, low-level disinfectant, option B, intermediate level disinfectant, option C, high-level disinfectant, and option D is used for fumigation. The right answer is option C, that is high-level disinfectant. Glutaraldehyde is an high-level disinfectant. It is a high-level disinfectant solution. What is meant by high-level disinfectant? High-level disinfectant, they have effectiveness equal to the sterilization. Equal to sterilization means they can kill even the spores also. Next question. Waste shops should be stored in option A, white translucent bag, option B, yellow color bin, option C, black color, and option D, red color bin. The right answer is option A, white translucent bag. Next question. How much percentage of Mcq'S should be used as nasal ointment for treatment in MRSA, infection? Option A, 6%, option B, 4%, option C, 2%, and option D, 8%. The right answer is option C, 2%. 2% of Mcq'S, it is used in the nasal ointment. It is used as nasal ointment for treating of MRSA infections. Next question. IPSG goals were developed in the year. Option A, 2010, option B, 2004, option C, 2006, and option D, 2008. What is meant by IPSG goals? That is International Patient Safety Goals. IPSG goals, they were introduced in the year 2006. Next question. The pH of SDA to grow fungi is. What is SDA? That is Sabouraud's dextrose agar medium. It supports the growth of only fungi. What should be the pH? Option A, 5.6. Option B, 6.8. Option C, 7.0. And option D, 8.2. Here the right answer is option A, that is 5.6. So slight acidic pH will be there. Next question. Leventhal medium is specific for option A, Pseudomonas, option B, Vibrio, option C, Neisseria species, option D, Haemophilus. The right answer is option D, Haemophilus. Next question. Cold sterilization is done by using option A, non-ionizing radiations, option B, ionizing radiations, option C, sunlight, and option D, infrared light. Here the right answer is option B, ionizing radiations. So ionizing radiations, it is called as cold sterilization. Why? Because there is no production of heat during the ionizing radiations. So if you see non-ionizing radiations, when we use the non-ionizing radiations, there is considerable production of heat. That's what it is called as hot air sterilization. So ionizing radiations, they are called as cold sterilizations. Next question, which one of the following bacteria is cell wall deficient? Option A, E. coli. Option B, Salmonella typhi. Option C, Mycoplasma. Option D, Treponema pallidum. The right answer is option C, Mycoplasma. So Mycoplasma is the cell wall deficient bacteria. So as there is no cell wall, they show the pleomorphism. Pleomorphism means a great variation in the shape and size of the bacteria or existing in more than one form. So Mycoplasma is the cell wall deficient bacteria. It shows the pleomorphism. Thank you. Please subscribe my channel, share and comment. Thank you.